Good afternoon. So I will present after Boris our bioinformatics core facility, and I will mainly try to, here you can see that we are really multitasking core facility, trying to do a lot of things in different areas, but the truth is that most of the time we are somehow collaborating with genomics because we're working mainly with genomics data, and all the things that we're trying to do is on sequencing bioinformatics, and we're trying to work with the sequencing data and give you some nice results, but also give you the ability to get these results yourself. I'll split my talk into these three big areas. So first I start with the main role of our core facility, which is the basic NGS processing, data storage, and taking care of what is produced basically. And there I'll just mention the big news, because I think that most of the users that we have already know how we work. And the big news is that we finally, through our collaboration with ICS, Maastricht University, we finally get to use the sensitive cloud that they were building for a few years already. They already got a certification that really is a very secure storage and computational resources with it. And for our users, the important practical step is that current storage will be changed, and even though all your data will be moved, you will now access it in different structure, different organization, and through different methods. Because so far, if you were a user, you might know that you needed to connect to some secure and dedicated VPN. Now this VPN will be changed for WireGuard, which is also just different thing, different VPN, but it's called WireGuard. And as I said, there's going to be another structure. Practically, here is a kind of schedule of all the groups that are currently have their data on the storage. So this is an approximate timing of the move of the data. I can now I prepared the data. I prepared this slide yesterday and then sent it. And today I had a meeting with the representatives of ICS, and I already know that it's going to be like 14 days behind this schedule. So I will update it on our website. So check out our website. There is a this new sign, and all the information, all the tables, everything that's is going to be there, including if you're just a researcher from some of these groups and you want to see if you would have access to the specific groups folder, you can have a look and you can find out. And also you will get the information when approximately this move will change. I hope that it won't disturb any of your work, because it should be really that it's going to be done during weekend, but also you will just going to be accessing your old folder, then it's going to be copied, and then at that point you will be said, okay, now start using the new storage, the new folder, and then only after that the old one will be deleted. And what I wanted to stress, the last thing, is that you might expect, because these WireGuards, unlike the OpenVPN previously, which was just general one, every user used just one. Now the WireGuard works in a way that every user needs to have a specific config file. So in collaboration again with ICS, hopefully in this next month and a half, you will get, if you're supposed to, you will get an email, looks like this, that you're getting the WireGuard VPN config, and then there's some instruction how to install it. Again, we will have all on our website. So if you have any doubt, either contact me or look on our website. So hopefully this move will happen. And now just briefly mention why we are doing it, and what I hope is going to happen next is that first off, already through this move to the sensitive cloud, since genomics data are inherently very sensitive data, especially the human genomics data are very sensitive data. So we are basically obligatory for us to move them to some secure space and work in the secure manner. But also in the future, since there are these quite a lot of both national and European level programs of open science. It was mentioned here already several times, these FAIR principles, open science clouds, but also on specifically genomics, there are these 1 million genomes projects. Everybody have the same goal basically of securely storing, but also providing the access securely to the genomic data across Europe. And we hope that with our collaboration with ICS, we are going toward that goal. So right now, we're working quite closely to get to have a federated EGA node here in Brno. It should be the first in Czech Republic, hopefully. We're discussing with Jarda over there. And hopefully be the first use case in Czech Republic for this genome data infrastructure project. And what it hopefully will bring you is the data security, as I mentioned, but also the accessibility of the data across Europe, but also very easily make your data accessible for publishing. Because I expect that, as we probably know, all these FAIR principles go toward that grant agencies will be forcing the researchers to actually publish data or they won't finance things. So through this process, through the infrastructure that we're trying to build right now, hopefully at the end, it will be very, very simple for you to just say, okay, this is accessible part and we want to have it accessible from that date on for this and this conditions, and we will just click and it will be published. So that's why we're moving our data, and that's what we're doing toward even furthering this endeavor. So that's one big part of our processing. The next big part is what Boris was talking. We got a new instrumentation, which means that we need to have a new ways to analyze the data. So there are these 3 instruments which are okay because they are all free-read. What Boris did not mention during his presentation, but was very much nicely asked, is about the long reads, about this P2 solo, which is the nanopore sequencer for long reads, and that's much harder actually to process, and it needs really a new type of bioinformatics that is now being developed across the whole world. So we are trying to participate, and in a specific custom project, I'll discuss a bit more about it. So that was the basic data processing. Now to the project specific support, which actually is probably what most of our users are like see us as what we are doing. It's not the majority of our works, to be honest, because the majority of our works is to keep things running and education. But basically, if you come to us with your problem, I have it here nicely, your problem is our mission. We really want you to come to us and ask your problem, and hopefully we can help you. But sometimes the problem is that it might take a while, which is something I want to address now to all the users, that from survey, in general, we have very positive results, and I'm pretty proud of it. Basically, the only problem mentioned several times was not ever the quality or the expertise, but the timing. And since I started as a head of the core facility last year, I must admit that there were some mistakes and some downtime. And since it was not only me starting as a head, but there were approximately 4 different changes in the personnel of our team. So when we have this nice graph, cookies graph of how teams perform, I hope that this year we follow this downside, and we're now in the upscale to performing, because really I can see now that our team and our procedures that are being placed are getting, team is getting tighter, procedures are getting better. So if you were somehow dissatisfied with the timing, bear with us, and hopefully we can make it better next year. So now to the more interesting stuff about specific projects and what examples of what we did or we like to do. We encourage you to give us the projects in this direction, because this is what we are very much interested in. So we have this new sequencer, and what it can do is to do the long-read sequencing, and it can sequence DNA, where the usage is usually the variant detection for medical genomics, but also the resolving of haplotype. It's very nice function of it, because with short reads you have never idea of because it's not long enough to really discriminate which haplotype it's come from. With long reads it's possible, and also for non-medical purposes, but for more interesting for plant research, research of these telomeric repetitive regions. Again, the long read sequencing could really help with it. And the whole other part is that it can really do the nanopore sequencing, could do the direct RNA sequencing. And as Boris said, we never did it yet here, but we're very much interested in doing it or analyzing the data that comes from it. And I will just mention that one of my colleagues already worked with the institute in US, which did the direct DNA sequencing, and they were testing. Basically, they have the cell lines untreated or treated with this 5EU modification. So it's not nature of occurring, but artificially induced RNA modifications. And then through this negative and positive set, he was able to create a machine learning model that is able to detect if the 5EU modifications are present or RNA from the sequencing data, and in what quantity, which now can be very nicely used. They use it for studying of the decay of RNA. But I also read multiple papers, and I'm very keen on trying the machine learning models that I know are out there for M6A modifications. So possibilities are out there, and yeah, I hope that we're going to get some of these projects. The other thing, just interesting thing I want to mention here is this spatial transcriptomics, which is basically a way to spatially resolve the transcriptomic levels on different spots of your slide. So for each spot, you know the transcriptome of the cells that are underneath that spot. And for that, in last year, again, with the help of my colleague, or did by Wojciech here, he did this very nice tool, Spatial Pass. You can access it, actually, it's really accessible. And it can help with the detection and analysis of interesting patterns on the spatial transcriptomics level. Again, if anybody would be interested in this direction, we would be super happy to collaborate on further development and help you with specific projects. The last slide, I put it there as an example of our work of custom analysis, is the slide I presented last Friday in Prague about the results from this 1,000 check genome project, ACGT. It's just a nice illustrative image of how you can use the sequencing results of the SNPs, the polymorphisms in each of the samples, to cluster the samples across the different regions in Czech Republic. So what I did was just a clustering of the samples based on their origin from the different regions, and it kind of worked, which I was not that surprised, but pleasantly surprised, I would say. So you can see it, Moravian regions cluster nicely together, the Prague with the central Czech Bohemian region also clustered together. Some are weird, and the Karlovy Vary is our kind. They're just different. So anyway, that was the kind of anecdotal experience what we had. So just very quickly about the courses that we teach. So we have 4 courses taught by personnel from our facility. 2 are more biological oriented, one in fall. They are basically the same course, one in Czech in fall semester, one in English in spring semester. And I encourage again your students to enroll. It's basically, this is the main thing. So how to plan, experiment, NGS experiment, how to look at the data, how to interpret the data. Then we have more bioinformatics oriented. So if you know or have people that are more focused and want to do the bioinformatics itself, there is this very nice course we started last spring, or it did not start it last spring, but we started teaching it last spring. And we are trying to really show you and teach you how to create an NGS workflow to analyze the NGS data. And then we have each semester, we have a bioinformatics seminar together with Faculty of Informatics when you try to invite interesting speakers to talk about bioinformatics. Other assistants, we definitely encourage you, if you want to hire somebody that would be on the sequencing bioinformatics side, I would really encourage you to contact us to help us with the selective recruiting process. I think it helps. Of course, we help with the IT infrastructure. I talked about it already. The community fostering, something I would really like to do. I did not have in the year so far. I did not have time or capacity to actually force everybody to like get together. But my idea is to, both from research group, but also from other core facilities, to connect the people who are more coding, because all the core facilities basically have some people who are the data analysts, right? And I got a feeling that if we get together once in a quarter of a year and discuss, we might find that there are some common problems that we want to solve. So that's something I would like to do in next year. Yeah, our acknowledgement, just check our website. I changed it a bit because it was just obtaining the data, and I got a feeling that if we don't obtain the data, you actually analyze them. But yeah, it's available on our website. So thank you for your attention.
Thank you, Wojciech. Any questions from the audience?
So concerning the direct RNA sequencing and analysis, do you talk about specific organism, or does it concern any organism?
I think it's general, because like it should not be organism specific. Like the detection of modification, because the modification.
No, not modification. RNA.
The RNA, no.
RNA sequencing.
It should not be like organism specific. You can.
But if
the modifications, some organism has specific.
Yeah, sure. Like it needs to be.
Molecule.
As I said, like you can detect, like regarding the modification detection, you need to have already pre-trained, like trained model to detect that specific modification. So, for example, this 5EU, they're like, well studied it. For M6A, I know there are at least 3 papers that claim that they have tools and models to detect it. I think that M6A is quite general modification that can occur across organisms. Of course, if it is some organism-specific modification, you would need first to have very nice data.
Yes, no, I'm not talking about the modification. Okay. I'm talking about, if you imagine that we would like to characterize again de novo, right? A structure of an RNA from an organism that is not so well annotated, and like a human genome, yeast genome, plants. Do you have the courage to try to go there to, or is it possible at all to?
What would be advantage of doing direct RNA compared to first cDNA?
Because we don't know how to approach it. We don't know about the processing of the RNA. We don't know how to process the samples.
Then, as Vojta said, I would prefer cDNA sequencing first,
which
Is
is
you convert it to DNA, have no modifications. So then you can use directly the sequence. The sequence, there could be errors, but at the end, if it's double-stranded, it could be
Yeah,
quite well corrected
Mhm,
even
mhm.
for long
Yeah,
nanopore
yeah.
sequencing.
So okay.
Then
Mhm.
it's
Yeah.
but it's cDNA, so you are losing
even
this
from cDNA,
modification.
the advantage of long reads is that you get basically the full transcript, which is super important if you either don't know or expect that there are going to be some changes, structure changes on the transcript.
So are you capable of analyzing such a cDNA if we don't know precisely the genome?
Yes, like you can compare to each other. You then basically do de novo transcriptome assembly, which we can do, right? But so and from long reads, it's much easier than from short reads because you basically have the full transcripts. So even if it's kind of complicated, which I expect in your case gonna be, that they're gonna be, like fragments and parts, it's much nicer because if you have at least one whole fragment of some RNA, which is somehow relevant, then all the fragments, and you have the whole read of the transcript, then all the fragments could be mapped to this read already. So you don't need to actually assemble it. You have like a full read reference, and then all these different weird things gonna be mapped to that. So I think that is definitely possible, and I would be very curious to try.
Yeah, we are trying to get there.